Yeah, thanks, Seb, for this kind introduction. It's indeed a great pleasure for me to be here and to tell you a little bit about polymer-based artificial cells. But before I'll start with that, I, of course, first like to congratulate you, Seb, on two things, I guess. First of all, for the completion of this very prestigious role you have as the Chair of Innovation Technologique. I think you've also put together a great symposium. I'm really looking forward to this day because I think we'll see really great speakers from all different areas that will cover this discipline. And of course, maybe a little bit prematurely, but I think you will survive one day. So also happy birthday with your birthday tomorrow. Well, I guess we'll have the dinner this evening also to celebrate that. And I think it's really nice to have kind of this combination of science and friendship. And I really appreciate our collaborations over decades and still continuing strong. And I hope we'll be able also to do this in the coming, at least decade, I guess. So, yeah. So, artificial cell research. Why are we interested in artificial cells? Well, this has several different reasons. For us, one important reason is kind of curiosity-driven. So, it is the question about how far can we push molecular cell assembly to create structures that mimic the hallmark of a supramolecular system, namely a living cell. And in order to do this, by doing this, we also hope to get better understanding about how, in fact, a living cell has originated or how biological processes are controlled, organized within a living cell system. And in fact, this research is not only purely based, I would say, on fundamental reasons, but it can also have quite a number of applications, because by developing systems that are lifelike, artificial cell-like, we can use these systems as simplified platforms, for example, in drug screening, but also in regenerative medicine as a kind of units that can instruct cells to become a certain type of tissue. So I think in that case, artificial cells have quite a lot of potential, both from a fundamental but also applied part of research. So what do we consider now to be kind of what lifelike behavior? Well, in fact, there are 5 criteria you could put onto this description of what lifelike behavior is. The first very important aspect is that you need some kind of compartmentalization strategy. In order to create something that can behave like a cell, you need to separate the content, the inside, from the outside environment and control the processes that go on over there. And it's not only about just creating the microenvironment, but also the architecture of such a microenvironment is really important. If you look at a eukaryotic cell, for example, there's lots of structure within the eukaryotic cell in the shape of polymersomes, organelles, I have to say, or a cytoskeleton that is present over there. Well, it's really characteristic of life is the ability to grow, divide, and replicate. And there are many activities going on in the artificial cell community that also try to mimic these aspects. Replication is still kind of a holy grail that is still far away, but growth and division, there are some examples already known that can achieve this. A very important aspect also of living systems is the ability to exchange information between each other and between the environment. And that's also typically something we like to also emulate within our artificial cell systems, where we can exchange molecules with either other artificial cells or with living cells to get communication pathways going. Metabolism is really an important aspect that people like to introduce in artificial cell systems to mimic living cells. And in this case also, the energy supply is important because living cells are out of equilibrium and you need to support them with energy in order to make them going. And finally, living systems are very good in adaptation, changing their conditions based on environmental pressure. And also that is one of the aspects we like to incorporate in artificial cells. Well, in our research, we specifically focus on compartmentalization, communication, and adaptability. And I will show you some examples about that. So our artificial cell platform that we've been working now the past couple of years is based on a so-called coacervate. It's a coacervate you get when you take 2 polymers of opposite charge, this positively charged amylose and this negatively charged amylose, and by mixing them together in water, they spontaneously phase separate in a droplet that is rich in polymers and an aqueous diluted phase that surrounds these droplets. And the reason we're interested in these kinds of coacervates is the fact that the crowdedness you get within such a droplet is pretty similar to the crowdedness you find in the cytoplasm of a living cell. So therefore you mimic really this internal structure of a living cell. The problem with coacervates compared to, for example, polymersomes or liposomes is that you don't really have a clear membrane and they're not really stable. So if 2 droplets meet, they will coalesce and they form, in the end, a macrophage separated system. So we found a solution for that by using this kind of polymer system. We normally make our coacervate slightly positive, and then this polymer has this negative anchor that anchors on the surface of the polymer of the coacervate, and this part of the polymer then stabilizes further these droplets from further growth, and then we have stable droplets we can use for further research. This membrane that we create is in fact a very interesting membrane because it's quite permeable and dynamic. And we've been able to show that by loading first the artificial cells with biomachinery, namely in this case enzymes. They get really up-concentrated very well, especially if there's a strong interaction force, in this case electrostatic. If you take negative enzymes, they go in really effectively. They have a 200-fold increase in concentration inside this coacervate phase. And because the membrane is permeable, we can add substrate outside of the artificial cells. It will go in, gets converted, the product can leave the cell, and go into a second artificial cell where it gets used for the second reaction that takes place. And we can follow this entire cascade by looking at the fluorescence of the final product. This is uptake by electrostatics, which is interesting, but it's also limiting because it's quite difficult to switch then this interaction because it's purely based on charge. But we can also make this interaction a little bit more sophisticated by using affinity tags to get cargo inside our artificial cells. And for that we explored this specific anchor, this nickel NTA group that is located over here. We connect this to an amylose, and this group interacts specifically with 6 histidines that are introduced in a protein of choice that is taken up. In this case, the protein doesn't have to be negatively charged, it can also be neutral, and it's taken up because of this specific interaction. And this gives us a very interesting opportunity, because if you now remove this Hest tag by having a cleavable site between the protein and the Hest tag, then this protein will lose its affinity and it will be secreted. And we've indeed shown that we can not only exchange small molecules between our artificial cells, but also proteins can be either taken up or secreted from our platform. So over the years, we have used this kind of platform for showing different types of functionalities we can have with this system, different types of lifelike behavior. For example, we can introduce motility in our artificial cells. By modifying the surface with an enzyme that is very active in converting chemical energy into kinetic energy, an enzyme like catalase, they can form these transient patches, and then they start pushing the particles forward, as you can see on this video. So here we can show an example of an artificial cell that moves around, showing motile behavior. We can also very well control the exchange of information between 2 populations of artificial cells by controlling the affinity. By having one protein located in the sender population, if we now remove the affinity for the sender population by this DNA exchange reaction, then this protein will be secreted and it will go to a receiver population for which this protein then attains its affinity. So we can very selectively move a protein from one population to the other in a very controlled fashion. Another example where we're going to go more into biological applications is where we study protein-protein interactions. So we've used this very important protein hub that is found in living cells, this 1433 protein, and we can indeed study the interaction with all kinds of guest proteins inside our artificial cell. And we can also use this for screening activities of drug candidates, so we can find out which kinds of small molecules have the ability to better stabilize this interaction, and that of course can now be done in a high throughput fashion, which means that we can develop this into a platform for effective, active compound screening. However, today I'd like to focus a little bit more on our work on kind of structural or architectural control that we have in artificial cells, and especially I'd like to address 2 elements, namely multi-compartmentalization, and the second aspect is the artificial cytoskeleton I'd like to discuss with you. So multi-compartmentalization is in fact something that started off our work in artificial cells, and I have fond memories of this project because it was a joint collaboration with Seb's group that we published about 10 years ago, involving 2 excellent PhD students, Mette Marguet and Ruud Peters. And Mette is in fact also co-responsible for the beautiful picture you see on the program, if I'm correct. So you see this beautiful illustration of artificial cells. I think Mette was also responsible for drawing that one. So this kind of was a nice way of collaborating. It's always good to talk about science when you're at conferences or when you're looking at posters. And in fact, at that time, we kind of parallel developed technology that led to this kind of research. So in our group, we developed, in that case, still polymeric nanoreactors, where we incorporated an enzyme in a membranous shell that allowed small molecules to go in and out. And in Seb's group, they developed a very interesting methodology to incorporate polymersomes in a larger polymosome structure. And then, of course, the idea was kind of born very quickly, is that we could now put our artificial organelles in this larger structure, this polymosome artificial cell, to create a multi-compartment system in which we could perform a 3-step cascade reaction in a very effective way. And indeed, we could show that by confocal microscopy. It's not a very fast reaction. You can see it takes about a day. It also shows the stamina of the PhD students involved because you have to stay a day behind the microscope. But in the end, we were able to really show the effect of having a cascade reaction going on in a multi-compartment system. And I think this is one still of the first examples of where a multi-compartment artificial cell was produced. So based on this concept, we thought about, could we also do a similar thing for our kind of coacervate-based systems? So in that case, we came up with the following procedure. Of course, we needed, again, our polymeric-based or polymosome-based nanoreactors, so protein-loaded polymosomes, that were then mixed in with the 2 amylose derivatives that form the coacervate. And then by adding it to a polymer for stabilization, we should get something that looks like this schematically, where smaller organelles are located into the cytoplasm of the coacervate, and everything is then secluded, surrounded by a polymer membrane, which is indeed a permeable structure. So how can we do this? Well, for the polymosomes, we devised a kind of a different strategy than we used 10 years ago. In this case, we made polymosomes based on this specific block of polymer containing a polyethylene glycol hydrophilic block, this caprolactone trimethylene carbonate hydrophobic block, which is highly flexible, doesn't have any internal structure, and therefore it's a very fluidic system that is also capable of letting molecules go in and out, at least small molecules. These structures spontaneously assemble into polymosomes, and we loaded them with 2 enzymes, kind of a classical cascade enzyme system, glucose oxidase and horseradish peroxidase. And we then measured whether these polymosomes could be used as artificial organelles by adding glucose outside of the polymosomes, and we could see that glucose could enter the polymosome and get converted by the enzyme inside. And if you then look at the kinetic parameters of these kind of encapsulated enzymes, we see that glucose oxidase outside or inside the polymosomes has more or less the same maximum rate. Of course, the Km value is slightly higher because that's based on the diffusion barrier that you've incorporated by putting them into a polymosome. In case of horseradish peroxidase, where the smaller molecule hydrogen peroxide has to cross the membrane, in fact, this Km value even is more closer in both cases. So we can make these polymosome nanoreactors, or artificial organelles, if you like, and now we have to incorporate them into a coacervate. And since the surface of these polymosomes is slightly negative, we could just mix them with our coacervate mixture that is slightly positive, and that led to the very effective uptake of these polymosomes within our coacervate, our artificial cell system. And here you can see kind of this image. It's like a Plantism painting, I would say, almost, where you see that you have 3 different kind of polymosomes labeled with 3 differently labeled proteins, a green, a red, and a blue label. You can nicely see they're nicely homogeneously dispersed within the artificial cell environment. You can here see also the 3 colors separately predicted. And this is kind of an outzoom picture showing that this is the case in all of our artificial cells. So does the system now also really work? So can we really perform a cascade reaction in this system? Well, indeed, it works very well. If you add glucose outside of the artificial cell, it can first cross the border of the membrane, then enter the artificial cell. It then can enter the first polymosome where the glucose oxidase is there. It gets converted, and the product then moves to the second polymosome. And we can then see that we have permeability and exchange of molecules from outside the artificial cell to the inside, but also within the artificial cell between different organelles. If you put the 2 enzymes together, then in fact the turnover is even higher than in case when they are separate compartments, which is, I think, a logical effect of diffusion. So these were structures that were made in a way that, in fact, we had a negatively charged polymosome that is kind of, well, taken up effectively within our coacervate system. But then, of course, we had the same idea that we had with our proteins, where we could control the affinity of the coacervate, of the cargo for the coacervate by changing the interaction between the 2 units. So in case of polymosomes, that would mean that we were interested to see if we could change the surface of the zeta potential of our polymosomes from slightly negative to positive, because if that would be the case, if you could create a positively charged polymosome, then we should lose the affinity for the positively charged artificial cell, and that then would lead, hopefully, to secretion of the polymosomes. If that would be the case, if you could achieve that, first of all, the question was, do these polymosomes then, well, can they pass the membrane of the artificial cell? But if you could achieve that, then, of course, it would look similarly to what we find in living cells, namely this exosome release. And especially this would be interesting if you could then release some kind of fragile cargo like RNA towards living cells. So the idea we had to achieve this is to take a polymersome where one of the blocker polymers was modified with this nitrobenzyl group, which is in fact a UV cleavable group, and after cleavage an amine group will be introduced. Of course, when you have an amine on the surface of such a polymersome, then the zeta potential will increase, and hopefully we create enough of this change to make them positively charged. So the idea is really to mix in first the kind of neutral polymersomes in the artificial cell system with our amylose derivatives, stabilize it with the topolymer, and then with UV light, we should then create this release. So first of all, we had to check whether it's possible to make these polymersomes charge of surface switchable. So we took different ratios of nitrobenzyl polymer with our regular polymer. We also modified a little bit the hydrophobic block by incorporating this imidazole, because this imidazole effectively interacts with siRNA, and that's in fact the cargo we'd like to deliver to living cells. And then by making this polymersome and by changing the ratio between the neutral polymer and the charged, well, the switchable polymer, we can see that at a certain point, indeed, we can nicely switch from a negative to a positive zeta potential without, in fact, disturbing the polymersome structure. So they keep the same polymersome morphology, as you can also see over here, and they don't change after UV switchable behavior, but of course the zeta potential quickly changes. If you then load them with an siRNA that is labeled with Cy5 dye, we can incorporate them now in artificial cells, and the red color is originating from the Cy5. So what happens now if we take our artificial cells and start to irradiate the sample? Well, then indeed we can see that we can get release. So in fact there are 3 experiments done over here. The first one is where we just take siRNA and mix this in with the artificial cell. The siRNA is negatively charged, so it gets in very well electrostatically. And of course, it's not released upon a UV irradiation because there's no change in features. If you take a polymersome that is not switchable, and we load this with Cy5 siRNA, also there we don't see any release, if you can also see kind of quantified over here. But if you now take our nitrobenzyl modified polymersomes and we irradiate them, we see that over time, indeed, our polymersomes are released from the artificial cell. And it's quite surprising to see that this goes so effectively, because indeed they have to cross this membrane, but somehow the artificial cell is accommodating enough to let these bigger particles go through and be released into the medium. So can we do this also in presence of living cells? Well, the first thing, of course, is to check how biocompatible the system is. Well, in fact, they're pretty compatible unless you go to quite high concentrations, especially some of the components in the coacervate can be toxic at high concentrations, but if you stay below these concentrations, in fact, the system is pretty compatible with living cells, in this case, HeLa cells. And if you then do the same experiment where we have non-activating polymersomes, or we have the activatable polymersomes, and we have the cells here in green and the artificial cells in red, and we start to irradiate, then in this case, we don't see anything happening, nothing is released, and the cells remain just green. But if you take the ones that can release the polymersomes from the artificial cells, here we see clearly uptake of the artificial organelles, the polymersomes, inside the living cells. And this is the quantification of that experiment. So this was just a release of kind of a dummy siRNA, but of course we would also be interested to see if we could really then also affect the biological function of our living cells. And for that we loaded this LAMP siRNA, LAMP1 siRNA, inside our polymersomes. It blocks this production of this specific protein, and we can nicely detect this with an assay that if the blocking happens, then this protein is not present, and then the particles or the cells will not become red. And in this case, we can nicely see that this works also effectively. So if we just have the cells, they just express LAMP1 as they should. If we then add our polymersomes, pre-made without the artificial cell, to the living cells, they are taken up effectively as RNA gets delivered into the cell, and we see a pretty effective knockdown of this protein. If you take now our polymersomes and put them in the artificial cell and we don't irradiate, nothing happens, so they stay in the same level of expression as the control, the cells, as without any treatment. But if you then shine light on it, we see that it's a constant decrease, a nice gradual decrease in LAMP1 expression, showing that we can indeed controllably release our polymersomes, our exosomes, if you like, from our artificial cell system to the living cells, thereby knocking out LAMP1 over time. And since it's a UV-controlled process, we can do this also in a spatio-temporal controlled manner. So here we take a slab of cells where we only irradiate this part, and then you can see that over here The red color is gone, more or less, while over here it still remains, showing that we have indeed this spatial temporal control over the release of our exosomes and therefore the deactivation of a certain protein in living cells. So this is a little bit about multi-compartmentalization. So the second part of my talk, I'd like to focus on our work on artificial cytoskeletons. And this is in fact the topic of interest of many researchers in the artificial cell community, and there are many beautiful examples that have been published over the past years. I just put a few of them over here. There's work done by the group of Petrashvili, where they incorporated natural cytoskeletons based on actin inside the giant unilamellar vesicle, and they could really show the formation of a cytoskeleton in there and also the deformation of the liposomes by the presence of this cytoskeleton in there. Now, other groups have worked on, for example, using DNA-based systems. Beautiful work from the group of Hest and Gottfried is that they made these DNA tiles that are also dynamic and that also allowed the transport of molecules along these fibers inside an artificial cell skeleton structure. And also quite recently work by the group of Ronnes-Freeman was done in which DNA peptide hybrids were used as a cytoskeleton. So the field is really actively moving forward to incorporate internal structure inside artificial cells based on these fibrous-like motifs. So when we started off, we also asked ourselves what kind of group can we then use, can we introduce? And in fact, we had an idea to use this specific building block, which is this diacetylene that is depicted over here, which is modified with two hydrophobic side chains over here, and then a polyethylene glycol group, and then a functional end to the chain. The reason we chose for this is that it's known that these kinds of structures can form hydrogen bonds between the urea groups, and that leads to a very effective stacking of these diacetylene peptide amphiphiles, or amphiphiles. The nice thing about the stacking is that if you stack them well, then these acetylenes get organized in such an effective way that upon irradiation with UV light, these two diacetylenes will start to polymerize, and they form a kind of a covalent bond within the fiber structure. Besides the fact that we can introduce for example, a negatively charged unit that allows uptake within our positively charged artificial cells, we can also introduce other functionalities, like azides or these cyclic alkynes that are depicted over here, which can be used for effective modification later on by click chemistry. That is a spontaneous reaction between the kind of orthogonal reactive groups. So it also gives a lot of freedom in modifying and tuning the functionality of these fibers. Well, this process works very well. So if you take diacetylenes, you irradiate them, they make this polydiacetylene, work already known by the group of Gerard Wegener in the 70s. So it's a very well-studied system, topochemical polymerization. And indeed, in the first instance, of course, you make a kind of nanometer-sized fibrils that are depicted over here. But since these fibrils are negatively charged, if you now add a positively charged polymer to it, they start to bundle up. And then you get kind of this aggregation of these small fibers into larger structures. And you can also nicely visualize that if you look, for example, with electron microscopy or cryo-electron microscopy in this stage, you see the individual kind of polymers. And if you then bundle them up, you need to go to confocal microscopy to see this pattern of bundles that is occurring within the structure. So what happens now if we add these different fibrils to the system? And then something, in fact, to be honest, we didn't really expect what happened afterwards, because we had two different types of fibrils. We had fibrils that were functionalized with acid groups and azides, and we had fibrils that were functionalized with acid groups and these DBCO molecules, these alkyne groups. And these alkyne groups are pretty big. They have two phenyl groups also in there, and they're pretty hydrophobic. And this has an effect on how the fibrils behave when you add them to artificial cells. Well, if you just take these azide-modified ones, they are hydrophilic fibers, so they just nicely disperse within the artificial cell environment, and they form kind of they occupy the entire lumen or the cytoplasm of your artificial cell. But if you now go to these more hydrophobic fibers, what happens then is that they don't stay inside, but they start to accumulate on the inside of the membrane side of the artificial cell. And you can nicely see that over here, that they're really interacting with the polymer membrane. In fact, it's even so that if we don't add the polymer membrane, which we can also do, then the fibrils will be just homogeneously distributed. So the interaction with the polymer makes these fibers move towards the surface of the artificial cell. And this now gives us quite an interesting feature, because we can, by controlling this functionalization, we can either kind of have interaction with the membrane with our cytoskeleton, or we can have our cytoskeleton dispersed within the lumen of the system. So what are the things we can do with this? Well, the first thing we can try, of course, is to see what happens if you put these, such a cytoskeleton on the inside of the fiber, of the membrane. And here you can again see with the STAD the positioning or the formation of these fiber structures. This is a 3D image. You can also see that it's not fully covering the entire surface, but you have areas where the fibers are located and also areas where they are not located. But what is the effect now of having this internal cytoskeleton network present? Well, it has an effect on the mobility of the membrane. I already mentioned our membrane is pretty dynamic. So if you do this fluorescence recovery after photobleaching experiment, we see that indeed in the case of the non-modified membrane, very quickly after that you bleach a part of the membrane, fluorescence gets recovered because of the dynamics of the polymers on the membrane. However, if our internal cytoskeleton is present that interacts with the membrane, this process is well, significantly slowed down. So we have either just the empty ones, that's a little bit like this, but then if you have a medium coverage or high coverage, we see that indeed the recovery becomes slower, and furthermore, it also is not complete anymore, meaning that we can immobilize parts of the membrane. And this is really interesting because it gives us opportunities to play around with the dynamics of the system and ensure that we therefore can also well control, for example, the interaction of molecules displayed on the surface with, for example, living cells in a certain way. So what about the other one? So if we have the hydrophilic cytoskeleton, well, of course, the azides, we didn't introduce them for nothing. We can use them for further functionalization. And one functionalization we can do, as I already mentioned before, is that we introduce this kind of nickel NTA group over here by just using a click reaction between a cyclic alkyne and the azide. So a spontaneous reaction that happens, and now our fibrils are functionalized with this NTA tag. So we have an affinity tag on the fibrils. So how can we use this? Well, we can now use this to introduce a scaffolding function for our artificial cells. So the cytoskeletons, in fact, well, bring molecules together on the fibrous surface that therefore can work together. And to show this, this concept, we use this specific type of system, where we have, in fact, a so-called split luciferase. A luciferase is an enzyme that can convert this compound by oxidation into light, so it creates luminescence. And this can easily be detected, especially when you connect it to a fluorescent protein that is activated when this light is produced, showing in the end green emission. This luciferase is known to be split into two parts, a large part and a small part. And if these parts are separated, of course the enzyme is not active, but if they're brought together, if they interact, then in fact the enzyme activity is restored. And of course this bringing together is based on proximity. So you need to bring these molecules together close enough to make sure that this interaction takes place. So if we take these two compounds and we just mix them in bulk, we see we have hardly any luminescence or fluorescence being created by these two loose compounds because their interaction is pretty weak, so they will not look for each other in the bulk phase. But if you take the same concentration of compounds and we add them to our fibrils, then we see that indeed they start finding each other. It's more difficult to see over here, but if you look really at the luminescence, you see there's a huge increase in luminescence, showing that we can now place the two parts on the scaffold, and therefore they are brought in close proximity, and therefore luminescence is restored. The enzyme gets restored. This also even is increased if we bundle the fibers by adding this positively charged polymer, and therefore there's even more kind of possibility for the two parts of the enzyme to come together and to become fluorescent. And even if we now put everything in our artificial cell system, we still see a very big increase compared to the bulk of our system. Slightly lower than this, and that of course has to do with the fact that this is a kind of, well, the diffusion in here is much slower than of course you have when you have a fiber just in, or bundle in solution. So this is about scaffolding, but of course a cytoskeleton primarily is also known to be very important for mechanical properties. And in fact, until now, this has not really been investigated in too much detail. So we were interested to see if having a cytoskeleton inside the artificial cell also would affect the mechanical properties of this artificial cell, make it more closely resembling to living cells. And that's very important because for effective functional behavior, you don't only need chemical signals, chemical interaction, but especially also mechanical features are crucial for systems to effectively interact. And so we did the following experiment, and this is done together with a group of Jochen Guck from MPI in Erlangen, where we looked at the mechanical features of our artificial cells. And rather than studying every artificial cell by cell, by, for example, using AFM technology, we could use that technology, which is called this real-time deformability cytometry. Basically, what happens is that you make a microfluidic device that's slightly tapered, and you push your particles, that can be living cells, can be artificial cells, through this duct. And because it's skewed a little bit, you see there's a flow, a shearing force, kind of occurring onto the particle. And the measure of deformation then tells you a little bit about the mechanical features of this specific droplet that you put in. So this is how it looks like in practice. So you start, of course, with a spherical particle, you push it through the channel, and then you see it becomes more like this boot-like shape because of the deformation forces that happen. And you don't do this, of course, for one particle, but you do this for many particles, and you get such a plot like this. And from this plot, you can then extract information about, especially certain mechanical properties, especially the Young's modulus, can be calculated from this value. And then if you do this in a way, you compare this with other systems, then in fact you see a very important aspect of having a cytoskeleton present in an artificial cell. So these are empty coacervates, so they more or less are, I would say, liquid-like droplets. That's how they behave. You can add another crowder that doesn't create a fibrous network like dextran, that doesn't really change the mechanical features. If you stabilize the membrane, what we do with this cytoskeleton that interacts with the polymer on the surface, in fact, also not much happens over there. But if we have our cytoskeleton inside, as you see over here, then indeed there's a significant decrease or change in mechanical properties. In fact, the material becomes more rigid, and in fact, this system now can be explained very well with an elastic sphere model, which people normally also use to calculate the mechanical properties of living cells. And if you can now compare our kind of artificial cell system with a living cell, then we see that in fact, with respect to the Young's modulus, these two have very similar mechanical properties. And this shows us that now we can not only kind of emulate kind of lifelike behavior in the sense of, kind of mechanical signaling, of chemical signaling, but we now also have a system in hand that we can use also to investigate mechanical features that are similar or closely resembling those of mammalian or living cells. And of course, that opens up many other perspectives in how to use these systems. For example, in the field of antigen, artificial antigen presenting cells. So with that, I like to finish, but just only with thanking all these people that have contributed to the research. Also, I'd like to thank people from CIMOCHEM for their contribution on the artificial cell cytoskeleton project, also the people from MPI. Of course, also special thanks for Bordeaux people from starting off this research on multi-compartmentalization. It was a very pleasant collaboration, and I guess we still continue with that. Of course, I'd like to thank the entire group, the funding agencies, and you for your attention, and I'm happy to answer any questions.